Joining us today is Fraser McCready, who is a PhD student in molecular genetics at the University of Toronto. In our last episode, you heard Fraser discuss his recent neuroscience publication. In this video, we'll find out about Fraser's other research projects, how his interests in neuroscience developed, and how students can develop their computational neuroscience skills.
So after you got this publication, what kind of projects have you been working on now?
My main kind of focus, I have two main focuses right now. The first is to continue kind of focusing in on what potentially could be going wrong at the synapse in terms of molecular interactions and like a signaling alterations that could be driving this phenotype. And what we've kind of focused in on is a process called activity-dependent translation, which relates to synaptic plasticity. It's really interesting because one of the gripes I think I have about the way that very introductory molecular biology courses are taught is you learn about what's called the central dogma, that you have your DNA, which gets transcribed into RNA. The RNA gets exported from the nucleus and then is translated into protein. And whenever you look at diagrams of how that happens, it kind of just shows an RNA molecule just floating out of the nucleus until it bumps into some ribosomes and then gets translated. And in reality, that's not really what happens. That translation step itself is very tightly regulated in terms of when and where those transcripts are actually made into protein. And in neurons, specifically, a lot of transcripts are actually held repressed and transported way out into the dendrites near synapses, and are only translated into proteins in response to some sort of signaling event at the synapse, right? And so it's a way that synaptic plasticity can occur. You think, a lot of these changes in synaptic strength that happen in response to activity happen very, very quickly, and they have to be very tightly spatially localized, right? If you're a neuron that has 1,000 synaptic connections, you need to change the strength of one of them. You need to have a mechanism for making sure that you're actually changing the strength of that synapse and not any of the other ones, right? So what actually ends up happening is that you have these signaling pathways that end up causing this sort of local translation of RNA that's localized near synapses, which make proteins that then feed back to that synapse and can change the strength of it in a very rapid and localized way. So one of our hypotheses is that, well, maybe what could be going on is that if you have messed up the signaling by these mutations in SHANK2, that kind of program of all of a sudden rapidly translating proteins near the synapse that feed back and change the strength could be just disrupted. And so what we want to try and do is identify what proteins are actually being made in response to these signaling events, and then how those translational responses are different in the SHANK2 neurons versus our controls. So that's a big experiment that I've been working on for a while now and trying to troubleshoot. And then the second part that I'm working on is kind of trying to build more on the function stuff that we were doing in the original paper that we published back in April last year. So we had some collaborators do patch clamp electrophysiology on our cells to try and find out how it is that they're communicating differently with one another. And where we've kind of tried to expand on that is by using what are called either multi-electrode arrays or microelectrode arrays, depending on who you talk to try and get an idea of how these neurons are functioning as networks, as opposed to just looking at the function of individual neurons themselves. So essentially how this works is we have these special tissue culture plates where at the bottom of each well that we're culturing neurons in, there's this grid of very, very small electrodes, like an 8x8 grid, that the neurons we plate in those wells can kind of grow and form connections over top of. And so what we can do is then record the signal that's coming out from each of these electrodes in this array and identify when neurons are firing at specific locations within this well. So you kind of get this map of where activity is occurring in the well and at what time. And you can see these events where what we call network bursts, where a bunch of neurons across the network will all kind of fire synchronously together. And so that's kind of like a really interesting phenomenon to me, because in order for these neurons to all be able to sync up with one another and fire together as a kind of cohesive network, that synchronization is dependent on these plasticity mechanisms, right? So you have two neurons that are sort of firing in sync with one another, and you have these plasticity responses that kind of reinforce that synchronization. And so if we are thinking that there's something wrong with our SHANK2 neurons in terms of plasticity, looking at this network behavior could kind of give us insight into how it is that these neurons are able to wire up differently with one another. So yeah, I'm really excited with that. with that. And we have some interesting collaborations. As I was saying earlier, the importance of these collaborations. One of the issues that we kind of run into is that these recordings generate a huge amount of data. I think, like, if I'm recording one 12-well plate of networks, we do recordings for 5 minutes, and we generate about 5 gigs of data from that one recording. And we do this, 3 times a week for 9 weeks, right? So you have this huge amount of data that needs to be analyzed. And us as being mostly wet lab biologists, there are some tools that the company that makes these devices has, given us to be able to do some analysis of that data ourselves. But really what we want to do is collaborate with more like computational neuroscience focused labs to try and extract more data out of these recordings and find out more what's going on. So we have some collaborations with a couple labs at the University of Western Ontario, being Julio Martinez's lab and Lyle Wheeler, who are working on trying to extract more interesting kind of information about how it is these neurons are all connecting with one another, and doing some computational modeling to try and simulate these networks that we're recording. Yeah, so I think that's kind of the other part of my project that I've been working a lot on, and we're getting some interesting results with that.
How did you find yourself in the field of research that you're in right now?
I've been interested in neuroscience for a very long time. It's actually kind of funny. I think what originally piqued my interest in neuroscience was this dumb book I read in high school that was kind of this, like, it was called The Holographic Universe. And it's kind of one of these books that tries to like give this, unified theory for how the whole universe works. And it's trying to argue that the universe is just a big hologram, right? It's a really bizarre book. It starts getting into things like paranormal stuff near the end of it. It's not very scientific. I don't want to give that impression, but one of the things it was talking about, and I remember reading, is, the fact that when you look at the world, it's not as if you just have some holes in your face that your brain like looks out of and is looking at things directly. Like, it's just your brain interpreting whatever electrical signals are coming in from your retina and your eyes, and that, this image that you see in front of you is kind of, I guess, just made up by your brain, right? Like in a very simple way of saying that, right? So I think that initial thing that I kind of read about, I think that was kind of like my first exposure to neuroscience, actually, was this weird book. But I thought that was like really, really, really interesting. And I always kind of wanted to pursue neuroscience since reading that, right? So I started my undergrad. I was actually enrolled at the University of Guelph in their psychology, brain, and cognition program. Did my first year in that. So it was like a psychology degree, but as a Bachelor of Science. And I thought like the psych courses were interesting, but I was finding that, the main, I guess, parts of that neuroscience that we were talking about that interested me the most was on kind of like the molecular side, and like what's actually going on within neurons that are, allowing them to, communicate with each other. So I ended up switching my degree over into molecular genetics, or molecular biology and genetics, with a neuroscience minor, and just kind of started working my way towards where I am now from that. my fourth year undergrad project was actually in the lab of Dr. John Vesey at the University of Guelph. And his focus was on looking at, I guess, neural precursor cells in the developing cortex and looking at mechanisms that kind of determine, oftentimes when these cells divide, one of the daughter cells goes on to become a neuron, while the other one will stay a neural precursor cell. And so we were kind of looking at, well, what is it that's different about the one cell that goes off to become a neuron from the one that stays a precursor cell? Like what kind of information is being passed on there? And actually, it's kind of interesting that the main kind of mechanism that we were looking at, and something that John had found out in his postdoc, is that I mentioned earlier the fact that there are RNAs that are localized to specific areas of neurons, and how that's a very tightly regulated process. So he had actually found out that in these neural precursor cells, there are certain RNAs that get localized to one pole of the cell versus the other, so that when that cell divides, only one of the daughter cells inherits these localized transcripts, right? Which might be encoding things like transcription factors, so that after that cell divides, those transcripts get translated into protein, and then those transcription factors go on and initiate some differentiation program that turns the cell into a neuron while this one doesn't. So I guess I'd kind of gotten exposed to neuroscience and stem cell stuff through that. And then when I joined my graduate program, I kind of wanted to continue on in that research, but maybe more in an applied direction. So adding on the autism research and the kind of the neurodevelopmental disorder research was interesting to me, because it's similar, but in a more kind of applied field.
Having done all your kind of studies with neural precursor cells in undergrad, did you feel prepared going into graduate school? What was the jump from undergrad to graduate school like for you?
I actually had a lot of different research experiences throughout my undergrad, to the point where by the time I was applying to grad school and then deciding to go there, I worked in a lot of different sort of situations with a lot of different types of research, and I felt fairly confident in my ability to do independent research. So I felt fairly confident about that. Obviously, it's always a big adjustment period whenever you switch between either high school and undergrad, or undergrad and grad school. So there were some, I guess, challenges to overcome with that, and sort of new techniques that you have to learn. For me, especially with the type of research I was kind of ended up doing with the RNA-seq analysis, with this sort of network analysis, I think learning to code became very, very important, right? Because the first time you open up one of these RNA sequencing data sets and it's 20,000 rows long, and you try to do any sort of analysis on it in Excel, it's just, you can't do it. So learning programs like R for data manipulation was really important. One of my committee members in my first committee meeting told me, you should start learning MATLAB, because that's how a lot of people who do electrophysiology work do their kind of data analysis. So I had to learn MATLAB, and those skills have become, I think, very, very important. So yeah, I guess if anyone is thinking about what can I do to make myself more prepared for graduate school, and you have the time to learn a programming language like R or MATLAB or Python, I think that's becoming more and more important in science in general. I think big data has become a big thing that's important in our lives now, and that's true for biological research as well. It's kind of inescapable. There's actually a big problem in molecular biology sort of research where people who do work with these sort of transcriptomics data sets in Excel, there's problems with genes like OCT4 is a big pluripotency gene. And when you open up OCT4 in Excel, it automatically converts that to October 4th, right? And so there's a lot of these different genes that you will go and you pull these published data sets from different like supplementary tables and stuff. And people will try to do these big like meta-analyses of things, and they're saying like, this is weird. It's like this is a stem cell paper, and I'm not seeing OCT4 anywhere, and it's because it's been converted to October 4th, right? So yeah, I think learning to use more programs is definitely beneficial.
Yeah, I think we've heard from some different people that the computational side of biology and neuroscience is getting really important nowadays, and about learning to program. Were there any opportunities like in your undergrad to learn that, or do you think it was mostly like something you have to pursue on your own?
The statistics courses that we took kind of included learning R as part of the course, but it wasn't implemented very well. It was sort of like overwhelming because now I'm trying to learn statistics and also learn this program at the same time. And it's like, I can't do my statistics homework because I don't know how to work the program. And, it was a little bit of a frustrating experience. I think, the thing that kind of helped me the most with being able to teach myself these coding languages was just taking one like first year intro to programming course, right? Because I think as long as you have an understanding of like, what is a variable, what is a loop, what is like an if statement and a for statement or whatever, like as long as you know those very basic tools, learning how to at least like scrape by in these languages is just kind of, learning the syntax of them or stuff. It's like it's just kind of like being able to go onto Google and type in like, how do I do this, right? And there's usually like an answer somewhere. But I think in order to even like be able to know what questions you need to ask, you need to have just a very, very basic understanding of like how computer programming works. Just like that first year intro to programming level is enough to, I think, eventually scrape by with things. I think I know like our graduate program is starting to actually require you to take a programming for life sciences course in your first year, or like one of the courses you're required to take kind of goes over it a bit, which is really great. I think that's a great opportunity for new students who are joining. Yeah.
That's some good tangible information and advice for anyone going into research.
Yeah.